Good morning. Thank you, Eric. Thank you to the organizers for having us here today. I feel really fortunate, actually, to be able to follow Dr. Musser. He really set up the framework for how we regulate substances in food. We're going to focus today on proteins, but I really think his introduction really provided nicely not just our approach to regulating proteins, whether they may be novel, or whether they may be changes in their use, or they may be through a new manufacturing, like cell-cultured food, but really all substances in food to ensure that their uses are safe. So as it relates to proteins as a subset of substances that are used or found in food for which we have the responsibility to regulate, the primary thing is that these substances are safe in food under their intended use conditions. And in our regulatory programs, both pre-market and post-market, safety is our ultimate goal and responsibility. And we have a myriad of programs to take a flexible approach that's geared towards a type of product and the process that may be used. As it relates to proteins, there's a long history of protein consumption upon which we can build our scientific knowledge and our approach from. We're not starting from new here. I mean, there's a lot of discussion today and a lot of use of the words novel and new and alternative, but when it comes to proteins, we have a long history of experience, both of them in the food supply, but also in terms of scientific information on their value and their role as a micronutrient, but then also the things about them that can cause safety concerns, potential allergenicity, toxic elements. These are things for which we have a lot of information. And as Dr. Musser also pointed out, fortunately, for us in the United States, our regulations are built in a way that we can understand that there is going to be continual innovation and evolution within the food supply, and our systems have long been responding to this. And so we have quite a long history of evaluating and watching, especially in the space of proteins, how their use, how their manufacture, how their appearance and how they appear finally in the food supply has changed over time and how industry chooses to use them. More recently, we're seeing a lot of increased interest, and there's a couple of examples that we have here, but I'm noting more and more, as I'm sure all of you are, there's more than one meeting a year where the topic of novel proteins, alternative food production approaches is really rigorously discussed. And while we talk about proteins, and we're talking about proteins here today as a specific subsector of substances added to food, really these represent a broad spectrum of types of products. And within the US, not all of these types of products come through one specific program. We use a variety of programs depending on the ingredient, how it's used, how it's been produced, and how it's expected to show up in the marketplace. And we heard Dr. Musser mention grass. I mean, some of these examples here, the traditional plant proteins, a new use of a protein that may exist, new manufacturing approaches to proteins that have long been used in the diet, new products of fermentation. These typically go through our generally recognized as safe notification program, and they're evaluated for safety based on their intended use through that program. But then we also have new proteins that can show up through potentially molecular farming products or whole foods from plants biotechnology, and here we have different programs. We have a biotech consultation program where we look at new species and varieties of plants for which proteins are certainly a part of, and also our cell culture program, our cell culture consultation program, which is our new program. And we'll talk a bit about each of these, but really the approach that we take to safety. And the thing that unifies all of these programs, which is the flexible approach based on what we know about the substance, how it's going to be used, and the data needs that drive that. One thing that is, I want to spend a moment to talk about through all of our programs, whether it may be a cell culture consultation program, a biotech consultation, GRAS notification, color additive, or a food additive petition, even food contact notifications, which were mentioned earlier today, is that we heavily engage with innovators in our pre-market engagement phase. So this is long before. We hope much early in a development process where industry is looking to expand the use of, develop something new, consider a range of questions that they may have so that we can engage with them to make sure early in their development they're asking the right scientific questions, they're developing the right sort of information, and even that they're coming in through the right program, right? I think, so like hemoglobin was mentioned. In that case, we went, you actually have two different uses here. You have use of something as a flavor and use of something as a color. Unfortunately for you, there's different legal requirements there, and you're gonna have to prepare to come in for both of those. Now, I don't think anybody really liked that news, but at least they knew early on that this was a legal requirement that they were going to have to deal with, and we were able through this pre-market engagement phase to be able to help direct them. We also think that this helps make our programs more predictable. Right? Now, safety is ultimately what we're going to have to look at, and the data will bear out in the end. But in terms of what to expect, we hope that while there's a lot of flexibility in these programs, this does help add a little bit of predictability and reliability in what your regulators are going to be asking for you. As it relates to pre-market engagement, we see there's two, I would like to segment out the two different types of use cases that we see here, because while safety is ultimately the goal and what our main responsibility and industry's main responsibility is, we are looking at two major differences in the use cases. And one of these is purified ingredients. So this is a scenario where a protein, in this case, as we're discussing today, is going to be used as a component in a finished final food product. It is not intended to be food in and of itself. It will be used and added to food as an ingredient or as part of food manufacturing. This is where we typically see things come in through the generally recognized as safe program, and examples would include new uses of traditional ingredients. We do know that there is a lot of interest here in taking things that may be considered an important ingredient in a minor a food that may not be in wide use, and taking that for some and expanding it into other different type of foods. And so that would be, we would consider a new use. Of course, there's also precision fermentation products, and then what we would consider molecular farming, which is expression of a protein through a commodity crop. These would be purified ingredients and could come through programs that way. And then we have two examples where we have what we consider, we would evaluate the whole food. And these would be our plant biotech programs and then human food from cultured cell animals. And we will talk about both of those. Just to spend a minute on our purified ingredients approach. Even within purified ingredients, we know that there can be a wide range of intended uses. And when we're thinking about purified ingredients, the intended use really becomes very critical in the safety evaluation, because it helps us understand and determine the expected exposure in the food supply, which any risk assessor knows is a really fundamental and critical part of that risk and safety assessment. For instance, we know macro ingredients are going to be a significant nutrient source, and they, depending on the foods that they're going to be used, are expected that consumers would have a much higher level of exposure to. And so when we're looking at the potential risk or safety impacts of that, we may have significantly more questions based on that elevated exposure use. Whereas processing aids could have little or hardly any protein present in the final product, and there may be significant steps within that manufacturing process that are going to deactivate or change that protein, so it may show up differently in the final food than when it was used within its original food manufacturing role. And so these are situations where we can really show that there's a big range here, and demonstrates the need for us to ask specific questions depending on what that intended use would be. And so this really approach allows us to take into account a total weight of evidence that looks at the identity, the intended use, the source of the protein, how it's eventually going to show up, to provide flexibility to ensure that appropriate analyses are being performed on the substance that's being evaluated under its intended use conditions. So I tried to explain here the degree of flexibility so that we're getting the right information so that we can firmly answer the question of safety. We're considering some specific factors. We've mentioned the source of the protein. We're also looking at the protein type and family. We know a lot about proteins. What sort of family does this come from? Is it coming from a known potential allergenic source? Does it have allergenic sequences? What will its dietary intake level be? What do we know about the biological function of the protein or these class of proteins? We're looking specifically at the amino acid sequence, especially in comparison with known allergens and toxins, to see if there may be specific questions or if this would raise concerns where we want to look further. Would there be follow-up testing that would be needed based on what's known in that comparison? We're looking at in vitro digestibility in simulated gastric fluids. We find this to be a really important tool. We need to know how this protein is going to behave in a biological system. As it relates to oral toxicity, what we've been finding, and we find is that we do receive oral toxicity studies, animal studies from time to time. Less and less, these are not being recommended by the agency unless there's a general case or situation where we see that they're needed. More often, we're using in silico approaches. We're using knowledge on information on protein family classes to really understand the potential safety profile, and we're not recommending specific animal studies in this case, and by that consider we're taking more of a NAMS-based approach here. And always we reserve the right on a case-by-case basis through this engagement. We're gaining knowledge and information based on the information that exists, the studies that are being provided, to ask for and request additional studies based on what we're learning. I want to take a minute to talk a bit about especially the purified ingredients as it relates to proteins specifically and animal studies and where we think their value may be. We really see based on this is an area that's really ripe for NAMs. I know that is a new alternative methods approaches. It's not necessarily the topic of this meeting today, but in general, the agency supports efforts to replace, reduce, and refine animal studies when this can be supported by the science. Ultimately, a reasonable certainty of no harm and safe food products is the ultimate goal here, but as it relates to proteins, we only request them when they're warranted based on the existing data and information. These are not a requirement before you engage with the programs for protein assessments. We know many proteins are routinely safely consumed, and that we know a lot about what we're looking for based on known digestibility and known protein sequence, and so we're really focusing on those elements within the review. Sometimes these are included in the weight of evidence, but what we've seen is they don't really end up being the really fundamental piece of data that really informs the safety assessment. Of course, it's always nice to have an animal study when it's available. No toxicologist is ever going to turn one down that I've seen. But generally, this weight of evidence approach is the approach we've been taking for protein safety, and so we recommend that animal studies be conducted on a case-by-case basis when dictated by protein function or the evidence such as in silico would suggest the need. So whole foods, I described within whole foods, we have a different approach because here we're not looking at the protein as a component of a final food. This is going to be the food in itself. And so we have two programs. The one is related to our plants biotechnology program. And here, when we're looking at the new plant, we are looking and taking a comparative approach. This approach, we're generally aligned with the Codex Alimentarius guideline to conduct food safety assessment for foods derived from recombinant DNA plants. This is the approach that we take. And in this approach, we're comparing the safety of the new food, the new plant variety, to those with the history of safe use, so the plant variety that it was derived from. We're looking at a compositional analysis. What's important to be aware of is that when we're doing this, we're assuming 100% replacement with the conventional counterpart. I think we all know and are aware that this is a widely conservative assumption, but for the sake of safety, we think it's supportable and an important one to take. This is especially important as we consider with our partners from the Center of Veterinary Medicine in components of feed, where often you will have one plant variety that is the majority of an animal's feed. And so we do assume 100% replacement, and we look at three main components. We look at molecular characterization to look at what those specific changes are. And we're looking at whether or not those new changes are going to result in a compositional analysis or change of those key nutrients, anti-nutrients, or toxicants. What is the effect of the change going to have on the potential safety of as it compares to that original plant variety? And then we're going to look specifically at the safety of the newly expressed substance. And when we're looking at the safety of the newly expressed substance, we're going back to thinking about the kind of elements that we would consider when we're looking at a protein as an ingredient. What do we know about that protein? Has it been in the food supply before? Are there any concerns for allergenicity? Are there any concerns for toxophore sequences? The ultimate goal is to assess whether the new food is as safe as the comparable food. If it were not, this is where we would raise concerns. All of our assessments for our biotech plant program, we have a whole inventory. These are available online with the analysis and our final letters, and the QR code is here for your convenience so you can easily find it, because our websites are not the easiest to navigate, I'll be the first to admit. As it relates to allergenicity, when we're talking about whole foods, especially in plants, there is a significant difference between the use of a protein as a purified ingredient versus the use of a protein as a component within a commodity crop, especially when it comes to allergenicity. And this is because when you're using a protein as an ingredient in food, labeling is an important component of its safety. Not only is it important to inform consumers, but especially within the US, for which I can speak to, this is what allergic consumers look for to make sure that they are protecting themselves. In contrast, and in whole plants, this is really, it's challenging to label a whole food in a way that would identify especially a new or unexpected allergens in food. And often within commodity crops, there's a lot of mixturing going on within the food supply chain. And so for this reason, when we're talking about new whole plant foods, it's especially important that we ensure any new proteins are unlikely to be allergens, and so this is a careful focus of the review here. So I want to talk a bit about our other program that looks at whole foods, and this is human food from cell-cultured animals. This is as I think we've all at least heard today, if you weren't already aware, the US has completed 2 such cell culture consultations so far. FDA has completed its portion, which evaluates the safety up to harvest of the cell material, and for these 2, USDA has completed its portion, which offers the grant of inspection after FDA has completed its safety assessment. This is a little bit different, and I think we've heard this from other speakers today, that the production process is critically important as part of that safety assessment. And so within the safety assessment portion for our cell culture consultation program, we're looking at the production materials, we're looking at the full process, and we're looking at the manufacturing controls to ensure safety along that process so that at the end, the final full food material we can ensure is a safe material. We're looking at the initial tissue collection and cell line development, maintenance and development of those cell lines, and the cell banking process to ensure that that original cell bank is safe and well identified. And then additionally, we're looking at the process to proliferate and differentiate those cells all the way through to the time of harvest, and that includes assessing the safety. At the final level, they would be in food for all components and inputs. So we heard a lot about the media and the process yesterday. We're looking at the components of those media, how much of those components are going to carry through to the final material to evaluate what we know about their safety and the exposure in that final level. We're focusing on the properties of the harvest cell material that are relevant for safety. And we're looking to make sure that we're identifying any potential unlawful food additives and other adulterants, and making sure that if there is the potential for an unlawful food additive, we are early in the process directing those submitters to the appropriate program so that can be dealt with. Right? There could be a path to getting approval even if you don't have something that's authorized. We can pursue authorizing that component through another one of our programs independently. And of course, I think we've already heard from Dr. Musser that we engage with USDA in consultations that involve livestock and poultry cells for which we have shared jurisdiction. So, as I've mentioned, we've completed 2 of these. So what are some of the elements that were included for these 2 that the FDA considered? And I mentioned some of these, but we'll repeat it because I think there's a lot of interest and it may bear worth repeating. So we're looking at the cell sourcing, where that original cell line derived from, what the species is, the identity, any modifications that may have been made to that original cell line, data that demonstrates the cell line stability, and then we're looking at the differentiation. Differentiated into what and how? We're looking at those. The cell bank generation process is important. There are specific substances that are used in cell bank generation that are important to consider. We consider these precursors. These are not used in the production process, but we need to know what they are to ensure that if they were to carry through at some low level, that would be safe. We're looking for a detailed description of the production process, including material inputs, information on the safety of those material inputs, the regulatory status of those, in addition to safety information where it exists. As part of this, because the production process is so critical to ensuring that you end up with a safe product at the end, we're looking at a hazard analysis, and we're considering what was the rationale and what steps have been put in place to ensure that hazards aren't carried through and they're appropriately controlled. And then finally, we're looking at the characterization and compositional analysis of that final harvested cell material. And we're looking at how it compares with a conventional comparator, but I just want to clarify, because I think especially we heard a lot about nutrition. We're using this as a reference point to look at the distribution of macro and micronutrients to make sure that if there's specifications for any hazard, that those are aligned with the comparator in terms of what some of those controlled specifications should align with, and then also to see how the production process results in what that final characterized food material is, so that we can track it over time and have that information to be able, as an identification piece. And then finally, what is the final conclusion about the safety of the harvested cell material? I want to talk a bit about transparency and information sharing. So all data that is critical for safety evaluation must be included within the publicly facing part of a cell culture consultation dossier. And the QR code will take you here to the 2 that we've completed, and you can see the amount and level of detail that is included in that public-facing dossier. In addition to that, we do receive supplemental information that we would consider trade secret that is not critical for the safety determination, and we can keep that information confidential. There is dialogue between the industry of what they may consider trade secret or very important confidential business information to make sure that the critical safety information is included in their public dossier, and that information that the agency thinks is necessary to us to fully understand and help validate their safety conclusion can be included in that supplemental information. And that dialogue happens at a pre-consultation phase. So this is just something to be aware of that adds to the process. So To wrap up, we certainly recognize and appreciate the recent interest in identifying new and alternative protein sources. Our ultimate goal and responsibility is safe food products, but we also recognize, and support the value of innovation in this space. It provides variety, can contribute to potential future food sustainability. There is certainly value in this space, and so we are taking a flexible and specific approach based on the process and the product and the intended use of the particular specific protein, so that we are able to support innovation. And we are deeply committed to increased transparency in this process. Throughout, you've seen some QR codes. I would encourage you to visit our websites. I would encourage you to engage with the industry early. Both of these are a way that we can support the work, but also increase the transparency in the work that we are doing. And you can find that information in the QR codes. So we do see innovation is going to continue. We do, as we've had now experience, had the benefit to be able to stand here and say, we do believe that our regulatory approach and our longstanding authorities, combined with our policy and scientific approach, provides the necessary flexibility and is adaptable so that we can appropriately regulate this and ensure the safety of these new proteins and production technologies. We know that there will be challenges, and each new application may bring unique questions, but we're prepared to stand and enable innovation in this space. So just a couple of resources to leave you with. We have 3 contacts, emails, one at pre-market. This would cover GRAS, color additives, food additives, even food contact notifications. It's really our catch-all box. If you're not sure, that's a good place to start. If you know specifically you're in a plant biotech space, you can reach out to our biotech inbox, or animal cell culture foods, you can reach out to our animal cell culture inbox. We're not as big of a group as I would like to be able to say we are, so we all know how to find each other. I encourage you to reach out, and we'll get you to the right place. So thank you for your time, and happy to take questions unless you want to wait.
Thank you very much.